November 27th, and I threw away the cat litter box culture just because it didn't grow anything interesting, like the smoked ham and the dog culture. I will show you the dog culture in a second. Dog culture grew a bunch of mold that I scraped off of it, and it also grew these little circular colonies right here that look like keeping everything in the refrigerator since I am trying to isolate psychophile bacteria. Okay, so as you can see, this is what I was talking about. So mesophiles are the majority of bacteria. They grow best at like human body temperatures, but there are also thermophiles which grow good at hot temperatures, and psychophiles which grow good at cold temperature. The reason I am keeping the bacteria culture in the refrigerator is because I am trying to isolate psychrotriles that like grow good at cold. November 27th, but I am going to allow the bacteria to grow for a few more weeks actually, because it's not as far along as I would think, or at least the smoked ham isn't. Disaster experiments here. So anyway, I have actually decided it is now December 1st, and I have decided to take the bacteria out since the cultures are beginning to be taken over by mold. And that means that I really just need to take the cultures out of the... In addition to what you already know, I'm going to transfer the new bacteria colonies onto these petri dishes using the sterile needle technique. Okay, so in addition to that, the cultures that I'm going to use are not like newly cooked up cultures or recipes. I've got these 3 bacteria cultures in my refrigerator that I cooked up a long time ago, and I just kept them in my freezer for a really long time frozen, and I figure I'm going to use them up. So yeah. Here are the 3 new cultures. I'm still frozen. And here is the dog culture V3. As you can see, after attempting to scrape off the mold, a lot of mold grew, but you can still see the coliform bacterial growth right there, the colonies. I'm going to try to isolate each colony and do a singular streak on the new culture. I'm going to streak it multiple times. As well, you can also see the pig smoked ham culture, 2016 smoked ham. This very small bacteria colony right here seems to be the best option. However, if I can find any other cool looking bacteria colonies, I am also going to streak them and streak the culture multiple times. And finally, there is one more thing that I'd like to show you. In place of the cat culture, which I threw away because it wasn't too interesting, I created this other, on the same day, my dad was cooking burgers with this venison deer meat, cut with, pork suet, which is, pig fat, taken from, the area near, the intestines of the pig, and, I took this meat, right, and I took a sample, and I have kept it in the refrigerator alongside these. Now, the meat protein acts as the protein source for this, basically what is a petri dish. And ever since I have taken the sample, the bacteria has multiplied, so that hopefully, you know how I streaked this with a piece of raw meat? Well, and didn't get too much bacterial growth, despite being raw meat? Well, this raw meat has been left to create more psychotrilic bacteria growth over the course of almost 4 weeks, just like these colonies, and I am going to take a sample, and that is what is the third culture is for. After this, I'm going to have to cook up some new petri dishes because these are the last frozen petri dish stuff that I made. It's not the new recipe, but the old recipe worked pretty good. This is like V3 or something. I related these two, dog culture V4 and smoked ham culture V2. But this one, this meat, I'm probably going to leave in the fridge for a longer period of time. For the simple reason that, there isn't really any mold growth on it, and I just kind of a lot of weight, you know. So hope you enjoyed, and see you in the next video.